And if you look at a villain TLR4 mice, all of these are tumors. And essentially, if you do a Swiss roll of their colons, the only epithelium they have left is dysplastic. They have wide swaths of ulcers, like this would be an ulcer. But between the blue arrows, hopefully you can see that, this is all dysplastic epithelium. Again, between the black arrows, this is all dysplastic epithelium. So what does survive our deassessing seems to be dysplastic. So, here's an intestinal monolayer. My bias is that TLR4 in health is expressed along the basolateral membrane. I have only a little bit of proof that that's true. And so that bacteria generally don't come in contact directly with TLR4 unless there's been a break in that barrier. But the interaction, if that does occur, and PAMPs then are recognized by TLR4, this results in the production of chemokines. KC would recruit neutrophils, CCL2 would recruit macrophages. So they recruit neutrophils and macrophages. These guys participate in the production of COX-2 and PGE2. This leads to EGFR activation by epithelial cells and promotes the growth of that tumor. So what does this mean for us as clinicians? Well, I think that in the future we will need to incorporate into our practices some amount of genetic profiling of patients of very distinct SNPs, depending on the question. I'm not entirely sure that genetics will ever be good enough to predict who is at risk for developing it. And why do that unless we have a really good way to prevent that person from getting it? But what I think is not crazy is to predict who's going to develop complicated disease or complications of their disease. And we're going to do that based on some kind of a genetic profile, some kind of an immunological profile, and of course always clinical acumen. So I'd like to leave you with the thought that although for right now a lot of our therapies are directed at a very late stage in the pathway, which are inhibiting TNF-alpha and inhibiting cytokines often produced by T cells, as we get more sophisticated, we might end up going to the very beginning of the cascade and trying to modify innate immune responses to treat inflammatory bowel disease. So this is my lab in Miami, although this is obviously not in the lab. They're having a good time. I normally come dressed up for work, so I don't actually get dirty in the lab anymore. And these are the people that I need to thank, both in Miami as well as people that worked with me in New York. And I left some really good collaborators in New York that I still keep in touch with. And for those of you that are grad students or have seasonal affective disorder, you're very welcome to come to Miami for a postdoc. So thank you very much for your attention, and I'd be delighted to take any questions that you have.
So for your villain TLR4 mice, the dysplasia that you see in the colon, do you think that's really related to an increase in the immune response that the animals are experiencing related to DSS, or do you think it's at least as much related to what seems to be a proliferative response that's there at the baseline, particularly if you said you saw that beta-catenin, more nuclear beta-catenin in sort of the resting state before you start to hit them with DSS? I wonder if you guys have any sense of what the balance is between those two factors, or if you're trying to get after that.
Yeah, I don't have the answer to that question. I will say to you that you can have wild amounts of proliferation that shouldn't lead to dysplasia, shouldn't make these cells look abnormal and lose their polarity. So I think they've taken at least more than just the proliferative hit. You can see part of our logic in checking the INOS was to see if they had a greater potential to generate reactive oxygen in other species that would then have a direct effect on the DNA. Well, I was talking to Julie yesterday, maybe I need to do sort of MSI stuff or look at other things that might be predisposing these animals. What is interesting is, are we mimicking that these animals are chronically infected? Like, does the animal perceive that it has a chronic infection? Do you see what I mean? Because even though we made a transgenic mouse that is transgenic for TLR4, I actually think it's behaving like a broad TLR overexpressing mouse. It's actually ligand independent, so I bet it's activating anything almost every other TLR would. So I think it's kind of an interesting setup to see what's happening as if the mouse is just thinking that it's infected all the way through. Yes, Vince?
So I guess kind of goes with the question, what do you think would happen if you were to rederive these villain TLR4 mice germ-free? Or, or very different, if you take them conventional and then douse them with
Antibiotics.
Ursulin's 4 or 5 antibiotic
Right.
cocktail.
Right, that's a great question. Well, did you read my competitive supplement to Obama? I asked Obama for more money so I could try to do that, because I think that's a really important question. Because actually one of the things that we've been sort of contemplating is maybe these animals develop these duodenal adenomas because they eat stool. And that it's not just the TLR4, it's also like they've sort of crossed some magical threshold of TLR signaling above which you get havoc, and that making them germ-free could address that. We are developing a germ-free facility, so I haven't just wanted to use the antibiotics because I figure I think that's a bit, I don't know, not perfect to give them antibiotics, but if I have to, that would be the next thing. The other thing I was going to do was cross them to TLR4 knockouts. I guess we could cross them to MYD88 knockouts. No, that wouldn't work. But to TLR4 knockouts, so that only the epithelium was making TLR4, and we got rid of TLR4 every place else. But I like the germ-free idea. You had another question, Vince? Or?
Well, people often equate the germ-free with the antibiotic treatment, but in my mind, they're very different because in one setting, the immune system grew up seeing
Right.
bugs,
Yeah.
and then you take that away,
Yeah.
and maybe that's why you have these funny results. Well, when you take that away, you take away the homeostatic, whatever the magical homeostatic signal is, versus having never seen
Yeah.
it and
Good point.
being raised without ever having
Good
that
point.
kind of stimulation.
And actually, that would at least we could do that right away too. We wouldn't have to wait to rederive them. So yeah, let me I'll write that on my list of things to do when I get back. Yeah.
Have you tried putting these mice on COX inhibitors to see if that stops dysplastic?
Yeah, that's a great question. The short answer is no, we haven't. I think that'll be interesting to see if that stops the duodenal adenomas. We should do that. For DSS, giving them COX-2 makes them very sick. That's not our work, and we haven't done that, but Balfour Sartor used COX-2 knockouts, and the animals, actually that in a way got us started thinking about COX-2 in the very first place, because if you read his results with a COX-2 knockout, it was almost like a phenocopy of the TLR4 knockout. And coincidentally, or perhaps not coincidentally, they also don't express COX-2. So we'll do that. That's a very clever idea. We will try that.